Hello everyone, welcome you all. In today's video lecture, we will discuss about next one point, that is evaluation of crude drug. In previous video, we had discussed about the quality, identity and purity of the drug in evaluation parameter. In today's video lecture, we will discuss about the different techniques, methods involved in standardization or evaluation of crude drug are as under. In that, first, organoleptic evaluation, second one, microscopic evaluation, third one, chemical evaluation, fourth one, biological evaluation, and fifth one, that is physical evaluation. These are 5 parameters or techniques included in standardization or evaluation of crude drug. Standardization of herbal drug is not an easy task, as numerous factors influence the bioefficacy and reproducible therapeutic effect. In order to obtain quality-oriented herbal products, care should be taken with respect to the proper identification of plants, season, and area of collection, their extraction and purification process, rationalizing the combination in case of polyherbal drugs. Standardization of the herbal drug is not an easy task, as numerous factors influence the bioefficacy and reproducible therapeutic effect. In order to obtain quality of the crude drug oriented herbal products, care should be taken with respect to the proper identification of the plant, then season and area of collection, their extraction and purification process. These are the factors including in the evaluation and standardization of crude drug. Then first one, that is organoleptic evaluation. Organoleptic evaluation, in that impression on the drugs. First one, that is organoleptic evaluation. In that, evaluation of crude drugs refers to the evaluation of a drug by color, odor, taste, size and shape. ऑर्गॅनोलेप्टिक इव्हॅल्युएशन मध्ये पॅरामीटर्स कोणते चेक करायचे आहेत आपल्याला? कलर, ओडर, टेस्ट, साइज अँड शेप ऑफ क्रूड ड्रग. Occasionally, the sound or snap of fracture and special features including touch, texture, etc. Organoleptic evaluation is also called as morphological or macroscopical evaluation. It is a technique of qualitative evaluation based on study of morphological and sensory profiles of whole drugs. The evaluation means conclusion drawn from studies resulted due to impression on organ of senses. Parameters used for this type of evaluation can be explained as follows. In that, first, color. The color is used in indicating the general origin of drug. त्या ड्रगचा कलर कोणता आहे? Example, aerial part of the plant. In that, green colored is. Then underground part, in that, devoid of green color. Aerial part of the plant कसा असतो? ग्रीन. आणि अंडरग्राउंड जो पार्ट असणार आहे तो कसा असणार आहे? डिवॉइड ऑफ ग्रीन कलर. So first one parameter in organoleptic evaluation, that is color. Then नेक्स्ट है size. The length, width and thickness of the crude material are of great importance while evaluating a crude drug. Procedure in that a graduated ruler in millimeter is adequate for the measurement of the length, width and thickness of crude materials. Small seeds and fruits may be measured by aligning 10 of them on a sheet of calibrated paper with 1 spacing between lines and dividing the results by 10. साइज त्या ड्रगची कशी आहे, त्याची लेंथ काय आहे, विड्थ काय आहे, थिकनेस काय आहे, हे काय करायचं आहे, सेकंड पॅरामीटर मध्ये चेक करायचं आहे. ओके. देन प्रोसिजर काय आहे, तर ग्रॅज्युएटेड रुलर इन मिलीमीटर इज ॲडिक्वेट फॉर द मेजरमेंट ऑफ द लेंथ, विड्थ अँड थिकनेस ऑफ क्रूड मटेरियल. स्मॉल सीड असेल, फ्रुट्स असेल, मे बी मेजर्ड बाय अलाइनिंग 10 ऑफ देम ऑन ए sheet of calibrated paper with 1 spacing between lines and dividing the results by 10. Then third one आहे odor and taste. In odor it can be described either distinct or indistinct depending upon amount of volatile principles in drug. त्या ड्रगचा वास कसा आहे आणि त्याची टेस्ट, त्याची चव कशी आहे हे सुद्धा आपल्याला ऑर्गॅनोलेप्टिक इव्हॅल्युएशन मध्ये चेक करावं लागतं. सो ओडर इट कॅन बी डिस्क्राइब्ड आयदर डिस्टिंक्ट ऑर इनडिस्टिंक्ट डिपेंडिंग अपॉन अमाउंट ऑफ volatile ऑइल or volatile principles present in the drug. Then distinct, that is distinct मध्ये aromatic, balsamic, spicy, fruity, alliaceous, that is garlic. Then camphoraceous, turbinathinate, that is turpentine. These are distinct odor. Indistinct म्हणजे when no such correlation is made. Procedure in that if the material is expected to be innocuous, place a small portion of the sample in the palm of the hand or a beaker of suitable size and slowly and repeatedly inhale the air over the material. If no odor is perceptible, crush the sample between the thumb and index finger or between the palms of the hand using gentle pressure. If the material is known to be dangerous, crush by mechanical means and then pour a small quantity of boiling water onto the crushed sample in a beaker. Then first determine the strength of the odor, that is none, weak, distinct or strong. And then color sensation, that is aromatic, fruity, musty, moldy or rancid. A direct compression of the odor with a defined substance is advisable. Example is peppermint should have an odor similar to menthol and clove have an odor similar to eugenol. Okay. सो हे काय आहेत, सेम ओडर असणारे एक्झाम्पल्स आहेत. पेपरमिंट शुड हॅव अन सेम ओडर और सिमिलर ओडर टू मेंथॉल अँड क्लोव हॅव अन ओडर सिमिलर टू युजेनॉल. ओके. ओडर आणि टेस्ट झालं. ओडरमध्ये डिस्टिंक्ट आणि इनडिस्टिंक्ट. इनडिस्टिंक्टमध्ये ॲरोमॅटिक, बाल्समिक, स्पायसी, फ्रुटी, ॲलियासियस, कॅम्फरसियस, देन टेरेबिनथिनेट, that is turpentine, and indistinct मध्ये when no such correlation is made. In the procedure of the odor and taste. Then taste. Taste, it is particular sensation excited by certain substance when brought in contact with special organs of epithelial layers of the mouth. Test can be of two types, true test and false test. In true test, that is acid, saline, saccharine, bitter, alkaline, metallic or tasteless. In false test, that is sensation to the tongue. In that mucilaginous, that is soft, slimy feeling or starch. Oily, it is bland, smooth feeling and linseed. Example is linseed. Then astringent, that is contraction of mouth tissue, that is catechu, cat. Then pungent, warm beating sensation, that is cinnamon. Acrid, that is unpleasant, irritating, coca. And noxious, that is induce vomiting. Example is ipecac. These are the false test and true test in organoleptic evaluation. Then fourth one, that is surface characteristics, that is texture and fracture characteristics. म्हणजे ऑर्गॅनोलेप्टिक इव्हॅल्युएशनमध्ये तुम्हाला सेन्सरी ऑर्गनचं आणि त्या ड्रगचं असलेलं जे काही मॉर्फोलॉजिकल आहे इव्हॅल्युएशन ते तुम्हाला काय करायचं आहे, आयडेंटिफाय करायचं आहे. म्हणजे त्याचं टेक्स्चर कसं आहे, त्याच्यावर फ्रॅक्चर्स किती आहेत, ते कसं दिसतं हे तुम्हाला आयडेंटिफाय करायचं. दॅट इज ऑर्गॅनोलेप्टिक इव्हॅल्युएशन. इन सरफेस कॅरेक्टरिस्टिक्स, टेक्स्चर अँड फ्रॅक्चर characteristics, examine the untreated sample. If necessary, a magnifying lens, that is 6x to 10x, may be used. Then wetting with water or reagents as required may be necessary to observe the characteristics of a cut surface. The texture is best examined by taking a small quantity of material and rubbing it between the thumb and forefinger. It is usually described as smooth, rough and gritty. ह्याच्यामध्ये तुम्हाला काय करायचं आहे, त्या ड्रगचं टेक्स्चर आणि फ्रॅक्चर्स चेक करायचं आहे. ओके, ते कसं असणार आहे, इट युजली स्मूथ, रफ और ग्रिटी. देन touch the material to determine if it is soft or hard, bend and rupture it to obtain information on bitterlessness and the appearance of the fracture plane, whether it is fibrous, smooth, rough or granular. For convenience, the description of macroscopic characters may be divided into 4 headings. In that, shape and size, color and external marking, fracture and internal color, odor and taste. These are macroscopic characters. Shape and size, color and external marking, fracture and internal marking, or internal color, and odor and taste. In shape and size, the size of crude drug encompasses measurements of length, breadth and diameter. It may be measured in mm or centimeter. Shape of the drug may be having different shapes. In that, sarsaparilla root, it is cylindrical, podophyllum drug subcylindrical, aconite drug conical, jalap ovoid and columba in the disc form. These are the shape of the drug having different shapes in cylindrical, subcylindrical, conical, ovoid, disc. In external color, the crude drug may acquire a variety of colors. The color of the crude drug vary from white to yellowish gray or yellowish brown, reddish orange or brownish black. The fruits and seeds have different colors, whilst barks possesses brownish gray to brownish black. The internal and external color of the barks varies, while internal color is little lighter than the outer one. These are external color. The crude drugs may acquire a variety of colors. The color of the crude drug vary from white to yellowish gray, yellowish brown, reddish orange or brownish black. The fruits and seeds have different colors, while barks possesses brownish gray to brownish black color. The internal and external color of the barks varies, while internal color is little lighter than the outer one. Fractures and internal color. It is fractures and internal color of the crude drugs refer to the external markings that the characteristics of a particular drug and color after breaking the crude drug. Internal color of crude drug is usually lighter compared to the external color. So example is turmeric or ginger. ह्याच्यामध्ये शेप आणि साईज आणि त्याचा कलर. कलरमध्ये एक्सटर्नल कलर ड्रगचा वेगळा असतो आणि इंटरनल कलर वेगळा असतो. काही काही ड्रग आहे त्याच्यामध्ये टर्मेरिक बाहेरून आपल्याला सुक्या असतं त्याच्यामुळे त्याचा लाईट कलर दिसतो, पण ज्या वेळेला आपण ते कट देतो त्याला त्यावेळेला ते आतमध्ये डार्क कलरमध्ये येलो कलरमध्ये प्रेझेंट असतं. देन according to the plant part used, all organized drugs can be classified into following groups for the study of gross morphology. In that underground plant parts in that root, rhizome, corm and bulb. Barks, woods, leaves, flower, fruit, seed. Study of gross morphology, it includes the visual examination of the drug. आपण बघितलं की आपल्याला समजतं ते सीड आहे, फ्रूट आहे, फ्लॉवर आहे, लीव्ज आहे की बार्क आहे, रायझोम आहे की रूट्स आहे. ओके, दॅट इज स्टडी ऑफ ग्रॉस मॉर्फॉलॉजी. दॅट इज इट इन्क्लूड्स द व्हिज्युअल एक्झामिनेशन ऑफ ड्रग. These drugs are classified into the following groups. In that barks, underground structures, leaves, flowers, fruits, seeds, herbs and barks. It includes all the tissues in a woody stem outside the interfascicular cambium which constitutes to the drug. Barks are collected from the trunk or branches of the trees in narrow strips. Example, cinnamon, cinchona, ashoka and kurchi bark. It includes all the tissues in a woody stem outside the interfascicular cambium which constitutes to the drug. Barks are collected from the trunk or branches of the trees in narrow strips. So example in that cinnamon, cinchona, ashoka bark, kurchi bark. Okay. Then underground structures. Underground structures मध्ये rhizomes, roots, bulbs, corm and tubers are the underground structures of the plant. They are swollen due to the storage of food material like carbohydrates and other chemicals. Example of underground drug that is ginger, turmeric and jetamansi. Powder drug possesses few microscopic characters but important microscopic characters for the identification. Particle size should not be less than 40. The broken cells, lignified walls, stomata, trichomes and cell contents. In that starch grains, allurans grains, calcium oxalate crystals, cystolith. These are the microscopic characteristics. Become evident in the mounted specimen. ज्या वेळेला आपण प्रॅक्टिकल परफॉर्म करतो त्यावेळेला त्याच्यामध्ये त्या ड्रगमध्ये असलेले लिग्निफाईड वॉल्स असतील, स्टोमॅटा असतील, ट्रायकोम्स असतील, सेल कंटेंट असेल, स्टार्च ग्रेन्स, अल्युरान ग्रेन्स, कॅल्शियम ऑक्सलेट क्रिस्टल्स, हे सगळे जे पार्ट आहेत ते त्या ड्रगमध्ये आपण ज्या वेळेला ट्रान्सवर्स सेक्शन घेतो आणि माउंट करतो मायक्रोस्कोप खाली, त्याला माउंट करण्यासाठी वेगवेगळे रिएजंट यूज करतो. त्याच्या अंडर आपल्याला त्या कॅरेक्टरिस्टिक्स दिसतात. The proper reagent to be used depends on the characteristics of cells tissues to be observed. Then example, starch बघायचं असेल, starch तर वॉटर अँड आयोडीन सोल्युशन आपल्याला ॲड करावे लागणार. जर आपल्याला लिग्निफाईड सेल्स बघायचे असतील तर त्याच्यासाठी आपल्याला फ्लोरोग्लिसरॉल H2HCl 1:1 प्रपोर्शनमध्ये ॲड करावं लागणार. कॅल्शियम ऑक्सलेट क्रिस्टल्स, लीफ इपिडर्मिस, ट्रायकोम्स observed under the chloral hydrate reagent. Then म्युसिलेज सेल्स बघायचे असतील तर पिंक कलरमध्ये आपल्याला रोदेनियम रेड हे रिएजंट ॲड करावं लागणार. ओके, these are all about the organoleptic evaluation. Okay, these are the first organoleptic evaluation. So आपण इथे स्टॉप करूया. Thank you everyone. In first part we'll discuss in today's video lecture, that is organoleptic evaluation. Happy learning, stay safe. If you have not subscribed to my channel, then subscribe first and press the bell icon to get the latest updates of upcoming videos. Thank you so much for fully watching this video. Thank you.